Hello, my name is Brianna Melanie, and I'm going to be sharing with you my research, which investigates the use of Ampelopsis brevipedunculata and Elaeagnus umbellata to produce biofuel as a potential New Jersey invasive plant management technique. I was inspired by this topic during a school project on New Jersey invasives and the current mitigation efforts surrounding these species. Upon noticing an invasive Japanese honeysuckle both near my house and school, I began investigating the known prevalence of invasive plants in the state and what could be done to reduce their detriments. Invasive species threaten ecosystems through various ways, such as habitat fragmentation and biodiversity decrease, both of which may lead to economic losses. This is demonstrated through the annual $137 billion spent on invasive species mitigation in the United States. One invasive species is Ampelopsis brevipedunculata, known as porcelainberry. An ornamental plant in the 1800s, porcelainberry is a vine that can be identified by its three-pointed leaves and its characteristic blue-purple berries. Today, it threatens native populations through its rapid growing tendencies and its ability to block sunlight and form thick mats on top of natives. Another invasive plant is Elaeagnus umbellata, known as autumn olive. Also an introduced ornamental in the 1800s, autumn olive is a shrub that can be identified by its silver-spotted red berries and green leaves with silver undersides. The shrub threatens native plants through its ability to reduce sunlight, along with its allelopathic abilities, which may lead to soil pH changes. Current methods that control these invasives include herbicides and mechanical removal. However, a novel application of yeast strains may provide a new outlet for invasive species control. Yeast is currently utilized to produce biofuel and bioethanol, which are used as sustainable substitutes for fossil fuels. Local areas may benefit from this novel application, as New Jersey is home to over 2,000 native species and 52 globally rare species. As now 39% of state flora is classified as at least exotic, the state suffers an annual loss of $290 million in potential profit for invasive plant control. The objective of this study was to determine the potential of porcelainberry and autumn olive for biofuel production and its application to New Jersey ecosystems. Furthermore, the ethanol yield of the species would be compared to determine its potential for the statewide utilization into biofuel. For this investigation, 5 test groups were created: sucrose, porcelainberry, autumn olive, black cherry, and blueberry. As pictured here, black cherry was collected at Site A and Site B in Ocean County, while porcelainberry and autumn olive were collected at Site C in Monmouth County. All specimens were stored at less than 0 degrees Celsius prior to testing. After collecting the specimens, I conducted Benedict's test to predict fermentative success. 1 milliliter of Benedict's solution and 5 milliliters of liquid derived from each test group were mixed in a test tube and placed in a hot water bath. Colors were analyzed to determine the relative presence of reducing sugars. Here is an overview of the fermentation process used for this study. Fermentation apparatuses were constructed to ensure anaerobic conditions. Distilled water, S-cerevisiae, and the test groups underwent fermentation for 2 weeks at 25 degrees Celsius before transferring to a stock container. This process was done 6 times per test group. Here is a more detailed construction of the fermentation apparatuses used for this study. A metal can was used to hold the fermenting batches. An airlock filled with water and a rubber grommet were included in each apparatus to prevent airflow. After all the stock solutions were collected, fractional distillation was used to extract ethanol from 50 milliliter samples. The ethanol would vaporize into the fractionating column and then liquefy while traveling through the Liebig condenser. 7 distillations were conducted per test group. Before I discuss the ethanol yields of the test groups, Figure 3 shows the results of the preliminary Benedict's test using the color changes of the reagent before and after the water bath. Porcelainberry yielded a bright orange color, so, due to the great relative sugar presence, it was predicted that porcelainberry would yield the highest fermentative success among the 5 test groups. It is important to note, however, that non-reducing sugars are not considered, as shown by the blank sucrose test. As for the ethanol yields, porcelainberry yielded the greatest ethanol yield of 20.34%, which was in line with the Benedict's test prediction. Additionally, the ethanol yield of autumn olive was 13.31%. An ANOVA test that compared the ethanol yields of the test groups yielded a p-value of 0.06, suggesting no significant difference as shown by Figure 4. This finding suggests that species like porcelainberry and autumn olive show potential for biofuel production comparable to more traditional sources, such as sucrose or sugarcane. The ethanol yields found during this study do not completely align with the Benedict's test predictions. For example, sucrose, while providing a blank test, still yielded ethanol during the distillation. Furthermore, Benedict's test suggested greater success with black cherry than blueberry, while the reverse was suggested to have happened. These suggest that the test groups may have also had non-reducing sugars, but the yeast may have been able to digest them through the production of invertase. This may be further investigated in the future to more accurately determine sugar presence in the fruits. Moreover, a particular finding of this study was that ethanol yields of both blueberry and porcelainberry were greater than 15%, which is the typical concentration at which yeast dies. This may be attributed to the gradual addition of ferment to the stock solution, a technique utilized by some craft brewers to increase ethanol concentration in their products. This technique may be utilized to increase ethanol yields in different biomass sources. I investigated the prevalence of porcelainberry and autumn olive in New Jersey using data from EDD maps. Porcelainberry was cited in 14 counties with the greatest densities in Cape May, Monmouth, Essex, and Morris counties. Most reports fell under the 2 to 10 plants per sighting density rating. Autumn olive was reported in each county. The areas of greatest concern include Warren, Hunterdon, Mercer, and Somerset counties. Most reports fell under the 11 to 100 density rating. As for the application of the ethanol yields, I conducted data analysis on the potential of these species for biofuel production. As shown in Table 1, the annual yields of porcelainberry and autumn olive may produce 50,000 and 60,000 liters of E10 fuel, respectively, the most common fuel with ethanol additives. For E85 fuel, which contains about 70% ethanol, approximately 7,000 and 9,000 liters, respectively, would be produced. These demonstrate the potential of the plants for biofuel for New Jersey organizations for local invasive management. Certain New Jersey counties may benefit more from this novel application than others. For example, counties such as Monmouth and Cape may have greater incentives for investing in this management technique. Figure 5 shows that certain counties have the greatest frequency of sightings in New Jersey. Furthermore, this type of invasive control may be advantageous over traditional methods such as herbicide application or controlled fires. Herbicides may not be wanted due to threatening surrounding plants, and controlled fires may be too large for smaller overrun areas. While invasive species show potential as biomass sources for biofuel production, a great deterrent regarding their usage stems from a lack of research regarding North American invasives and economic concerns regarding production. However, rising concerns over invasive species may increase sentiments towards invasive-based biofuels. Through this process, a variety of plants may be used, allowing for different areas to customize fermentation according to nearby invasives. Loyola University Chicago recently utilized invasive Phragmites australis as biomass for biofuel, displaying the customization possible through this application. The use of invasives may reduce competition of current prevalent food crops like sugarcane. Furthermore, it may limit the naturalization and invasion of biofuel specialized plants. For example, various specialized biofuel crops in Hawaii have become invasive due to overproliferation. As shown to the right, biofuel crops were more likely to have a high risk of becoming invasive than other introduced species. It is important to reduce the introduction of more invasive plants, considering that there are already species that can be utilized for biofuel and are already threatening local ecosystems. Measures that decrease transportation and machinery costs and increase biofuel efficiency through using cellulosic material can increase incentives towards this novel application in New Jersey and similar areas. The objective of this study was to determine the potential of porcelainberry and autumn olive for biofuel production as a management technique. The findings of this research support the use of these species for this cause. Future research should be focused on reducing production costs and increasing prevalence of biofuel production. In the future, I plan on creating biofuel using the invasive-derived ethanol and comparing fuel efficiencies. I will continue to investigate the potential of the lignocellulosic mass of these invasives for ethanol yield through designing chemical and enzymatic treatments. Furthermore, I plan to share my research among local strike teams to better determine the feasibility of such invasive control methods. Here is an overview of my presentation in poster format. Thank you for following along with this presentation. Also, thank you for extending to me this amazing opportunity to present at this year's New Jersey Academy of Science Research Conference. Have a great day.